In approximately 97% of all ALS cases, misfolded TDP-43 proteins clump together to form inclusion bodies that choke the cytoplasm of motor neurons. For decades, scientists faced a severe diagnostic limitation. This definitive molecular pathology could only be verified under a microscope after the patient had passed away. This created a massive roadblock for clinical trials. Without a way to look inside a living brain, researchers couldn't run in vivo assays to prove experimental drugs were hitting the right targets. This diagram shows a first-in-class PET tracer from AC Immune called ACI 19626. It is engineered to cross into the central nervous system and bind directly to these misfolded TDP-43 targets. Once bound, the tracer emits a signal that allows clinicians to map the exact spatial distribution of the pathology across the cortex and the spinal cord. This provides doctors with a highly accurate, quantifiable measurement of the total TDP-43 burden inside a living patient. By quantifying this protein load in real time, clinicians can now track the physical progression of TDP-43 pathology in parallel with the patient's observable symptoms. While PET imaging tracks the physical location of the aggregates, researchers at the Target ALS Consortium are developing fluid biomarkers to track the functional damage they cause. This biological diagram of a motor neuron illustrates the upstream failure. Under disease conditions, normal TDP-43 abandons its post in the nucleus, where it normally splices RNA, and migrates out to pool in the cytoplasm. Without TDP-43 there to edit the genetic code, rogue sequences called cryptic exons slip into the unedited messenger RNA transcripts. When the cell translates this flawed RNA, it produces entirely new, stable protein byproducts. Two specific examples are the cryptic proteins HDGFL2CE and IGLON5CE. Because these specific cryptic proteins are stable, they survive long enough to be secreted by the neuron, eventually crossing the blood-brain barrier and entering the cerebrospinal fluid. Their stability and ability to reach the spinal fluid make these peptides ideal targets for new, non-invasive biomarker assays. Because these specific proteins are only generated when TDP-43 fails to splice RNA, finding them in a patient's spinal fluid provides a direct, quantifiable readout of the protein's loss of function. This clinical visualization illustrates a major flaw. Aggressively dissolving TDP-43 aggregates clears the cytoplasm, but the surrounding neuron still collapses and dies. Clearing the clumps is useless if the native protein remains dysfunctional. The cell absolutely requires active, working TDP-43 to survive. This linear map of the full-length TDP-43 protein isolates a key discovery, a specific sequence within the prion-like domain called the conserved region. This sequence acts as the engine for liquid-liquid phase separation, ultimately driving the neurotoxic clumping. To counter this, researchers identified a small molecule called XL20 that binds exclusively to this conserved region. Because XL20 binds with such high precision, the protein's RNA recognition motifs remain completely unblocked and fully capable of performing their normal cellular duties. This structural stabilization triggers a critical secondary effect. It lowers the toxic accumulation of TDP-43 inside the motor neuron's mitochondria. By clearing the mitochondria, the cell restores its bioenergetic output. In A315T mouse models, this intervention successfully extended the animal's survival. Targeted structural stabilization, rather than blunt clearance, successfully rescues the cell's energy supply without destroying its ability to regulate RNA. Researchers in the Shawna lab have identified an additional complication, a truncated isoform called STDP-43, which is highly concentrated inside vulnerable motor neurons. Looking at this structural comparison, you can see the full-length protein on the left and the short isoform on the right. The short version contains specific C-terminal clusters called steric zippers that drive rapid, aggressive aggregation. To stop this aggregation, the lab mutated these zippers to physically force the protein out of its clumped state. The immediate intended result was achieved. The mutated short isoform achieved total solubility. But this created a biochemical paradox. The hypersoluble protein now binds to RNA transcripts with excessive, lethal avidity. This causes a catastrophic shift in the transcriptome. The neuron goes from including cryptic exons to aggressively skipping healthy exons. These missing sequences are known as skiptic exons. Stripping them out of the messenger RNA is just as lethal to the motor neuron as the original protein aggregates. Breaking aggregates is inherently dangerous if the process unleashes a hyperactive gain-of-function RNA binding state. This poses a strict biochemical challenge. Researchers need a way to prevent the short isoform from aggregating without pushing it into that hyperactive binding state. One novel solution is the use of short RNA chaperones, specifically a molecule known as CLIP34. This molecular model illustrates the mechanism. Instead of mutating the protein itself, the CLIP34 RNA chaperone binds directly to the 3' untranslated region of the transcript. This binding stabilizes the entire complex, physically preventing those steric zippers from locking together and aggregating. Because the protein is stabilized by a chaperone rather than permanently mutated, it maintains its native, safe physiological binding limits. RNA chaperoning sidesteps the gain-of-function trap entirely. It modulates the protein's native properties without abolishing them. If the upstream TDP43 protein cannot be stabilized, downstream RNA damage must be manually patched. This is the approach taken at University College London. They focused on a gene called UNC13A, which maintains neuronal synapses and is universally compromised when TDP43 fails. To fix this, they use antisense oligonucleotides. A synthetic ASO strand acts as a precision patch, aligning with the mutated UNC13A pre-RNA. It attaches to the cryptic splice site, creating a steric block. The splicing machinery skips the cryptic exon, generating a healthy transcript. These molecular patches functionally decouple the survival of the synapse from the failure of TDP43. The neuron can maintain its connections, even if the upstream proteins continue to aggregate. UNC13A is one of many targets. Here at Harvard Medical School, parallel rescue pathways are showing similar promise. Researchers are repurposing statins to modulate the mevalinate pathway. This triggers a repair response that successfully restores levels of STMN2, another critical protein normally lost when TDP43 fails. The field has moved past the helpless observation of terminal pathology. Researchers are now executing active, multi-level biological engineering. First, diagnostics. Clinicians can now see the disease working in vivo using PET tracers like ACI19626 and quantifiable CSF proteins. Second, upstream stabilization. Using small molecules like XL20 and RNA chaperones, scientists can prevent toxicity without triggering lethal gain-of-function splicing errors. And finally, downstream rescue. When the primary system fails, researchers can manually patch the resulting RNA errors using ASOs and pathway modulators. The ALS drug pipeline is shifting away from a blind search for aggregate clears and moving toward a targeted matrix that addresses both the physical protein and its genetic consequences.